Hello everyone, today we will talk about the glycolipids. So before moving ahead, let's first recall the classification of lipids. So lipids are classified as simple lipids, compound lipids and derived lipids. Simple lipids contain fatty acid and alcohol. Compound lipids contain fatty acid, alcohol and an additional group. And derived lipids are hydrolysis product of simple and compound lipids. In simple lipids, example of simple lipids are triacylglycerol and waxes. Compound lipids are phospholipids, glycolipids, and lipoproteins, and derived lipids are fatty acids, esteroids, eicosanoids, fat-soluble vitamins, and ketone bodies. In previous lectures, we have discussed about the simple lipids, that is triacylglycerol and waxes, and in compound lipids, we have discussed about the phospholipids. In this lecture, we will talk about the another compound lipids, and that is glycolipids. So let's talk about the glycolipids. Glycolipids, it's made of two words, glyco and lipids. So glyco stands for sugar. So when lipid molecule are attached with one or more sugar unit, they are called glycolipids. So they consist of fatty acid, alcohol and sugar. On the basis of their backbone alcohol, they are of two types. First one are glycoglycerolipids and second one are glycosphingolipids. Glycoglycerolipids contain glycerol as backbone alcohol and glycosphingolipids contain sphingosine as backbone alcohol. Glycoglycerolipids are most common in plants, algae and cyanobacteria, and glycosphingolipids are most common in animals. So basically glycolipids face the extracellular environment and they are present on outer leaflet of the plasma membrane. So this is the cytoplasmic side and this is the extracellular side. So basically this is the sugar part of carbohydrate part of this glycolipids and this is the lipid portion. So always this sugar part, carbohydrate part face the extracellular environment and they are also present on outer leaflet. This is the inner leaflet which face the cytoplasmic side and this is the outer leaflet which face the extracellular environment, and glycolipids mostly present here. What are the functions of glycolipids? They are involved in cell-cell recognition, cell adhesion, cell signaling, and they are also receptors for toxins and pathogens. So let's first talk about the glycoglycerolipids. So here the backbone alcohol is glycerol, and glycerol is 3 carbon containing alcohol. So here first and second carbon are attached with fatty acids, and with third carbon here the carbohydrate is attached. So this is glycerol backbone. Here 3 carbon are present and OH, 3 OH group are attached. With first and second OH group, carboxylic acid of fatty acid chain interact, and here sugar molecule is attached with third OH group. So this molecule is called as monogalactosyl diacylglycerol. So monogalactosyl means single galactose unit is present. Diacyl means 2 fatty acid chain are present, and this backbone is made up of glycerol. So this is the example of one of the glycoglycerolipid which is abundant in thylakoid membrane of plants, algae and cyanobacteria. So this was all about the glycoglycerolipids. They are mostly present in photosynthetic organisms. Next one is glycosphingolipids. Glycosphingolipids. In glycosphingolipids, the backbone alcohol present is sphingosine. It is 18 carbon containing alcohol. Here the first and third carbon are attached with hydroxyl group and second carbon is attached with amine group. And with this amine group, carboxylic acid of fatty acid chain interact with the amide bond. So when fatty acid interact with this sphingosine, this newly formed molecule is called ceramide. So basically glycosphingosine is this. It can be represented like this. So we represent sphingosine as inverted L, and here the fatty acid is attached and sugar face the outer leaflet extracellular environment. So this is the representation of glycosphingolipids. Let's talk about the classification of glycosphingolipids. There are 4 different classes of glycosphingolipids. First one is cerebroside, second one sulfatides, third one is globoside, and fourth one is gangliosides. Cerebroside is made up of ceramide plus monosaccharide. So here the sugar present is monosaccharide. Second one are sulfatides. They are made up of ceramide, monosaccharide, and one more additional group is sulfate. In globoside, here sugar molecule is oligosaccharide, and here in ganglioside sugar molecule is oligosaccharide, and one more additional group is present, that is N-acetylnuraminic acid. So if we talk about the ceramide, it is sphingosine plus fatty acid. So that part is common in all. How they are different? They are basically different in their composition of sugar. So they all have different sugar units. So let's first talk about the cerebrosides. They are made up of ceramide and monosaccharide. There are 2 types of cerebrosides on the basis of their different sugar unit, glucocerebroside and galactocerebroside. In glucose cerebroside, monosaccharide unit present is glucose, and it is found in plasma membrane of non-neural tissues. And second one, galactocerebroside, here the monosaccharide unit present is galactose, and it is mostly present in plasma membrane of neural tissues. Next one are sulfatides. Sulfatides are also cerebroside because they contain the monosaccharide unit, but they have additional group, and that is sulfate, and because of that this sulfate, they are negatively charged. So they are also called as sulfated galactocerebroside. They are cerebroside because they contain monosaccharide, and since here monosaccharide unit is galactose, they are called galactocerebroside, and since this galactose is attached with sulfate group, they are called sulfated galactocerebrosides. And because of this sulfate group gives it is negatively charged, because if you see the other cerebrosides were neutral, they were uncharged, and they are abundant in myelin, which insulate the axon. Next one are globosides. Here the sugar unit present is oligosaccharides. So they are also neutral, uncharged, and since they are oligosaccharide means they have more sugar unit present. More than 2 sugar units are present. One example of globoside is they are present on red blood cell membrane, and they are determinants of ABO blood group. So here the example of ABO blood group. So if you look at the O antigen, so basically this carbohydrate part act as antigen. So in O antigen we have these 5 sugar units. GLC stand for glucose, GAL for galactose, GAL-NC for N-acetyl galactosamine, again this is galactose, and FUC for fucose. This is the basic carbohydrate unit of O antigen. If you look at A antigen, it is similar to O antigen. All these 5 carbohydrates unit are exact similar as O antigen, except for this last one, that is N-acetyl galactosamine. And next one are B antigen. They have also similar, these 5 sugar unit are exactly same except for this last one, which is galactose, and because of this difference in their sugar unit, they act as antigen. Next one are gangliosides. Gangliosides are similar to globoside. They also have more sugar units, but they have one more group, and that is N-acetylnuraminic acid, which is present here. First sugar is glucose here, second is galactose, then it's N-acetylnuraminic acid. And this is the example of, this is GM3 gangliosides. Next one is GM1 ganglioside. It also, they have different sugar composition. Here only glucose, galactose were present. Here glucose is present, then galactose, again N-acetylnuraminic acid is present. And then it has another sugar unit, and that is N-acetyl galactosamine and galactose. So here GS stands for gangliosides and MS stand for mono. So let's first talk about this NANA, N-acetylnuraminic acid. It is also called as Nu5Ac. It is a type of sialic acid. What is sialic acid? It is family of 9 carbon acidic sugar molecule. So in human, most common type of sialic acid is N-acetylnuraminic acid, which is present here in both these ganglioside, in GM1 and GM3. So here the M represent, M means mono. If it's D, it is di. If it's T, is tri. So if single sialic acid is present, it is called as monosilo. If 2 sialic acid are present, it is called disilo. Here this is the example of monosilo, because here only single N-acetylnuraminic acid is present in both this GM1 ganglioside and GM3 ganglioside. And 1 and 3, these numerics represent their different mobility on chromatographic plate, and because of that they are named GM1 and GM3. They have different mobility because they have different structure. So they are abundant in nervous tissues, and here this is the structure of N-acetylnuraminic acid. So basically this is 9 carbon acidic sugar. This is 9 carbon sugar. Here 5th carbon is acetylated, and on this first carbon carboxylic acid is present, and at neutral pH this is this has negative charge, and because of this ganglioside are negatively charged. So cerebroside and globosides are neutral glycolipids, whereas sulfatides and gangliosides are negatively charged. Gangliosides are negatively charged because of N-acetylnuraminic acid, and sulfatides are negatively charged because of sulfate groups. So that was all about the glycolipids. Thanks for watching. See you in the next video.